I'm an associate professor here at the Department of Chemistry and Biochemistry. I run a research lab in this department, primarily focused on the immune system. We'll talk a little bit about that. I also serve as the director for the BioCenter, the Biosystems Networks and Translational Research Center, which is a virtual center that includes faculty here at SDSU as well as USD and School of Mines and Technology. I got here in the fall of 2008, so I'm coming up on a decade now.
Wow.
I was at the University of Michigan, where I spent almost 10 years in different roles, as a graduate student for a while, a postdoc, and then research faculty in the medical school. And then it was really my wife's choice about where we got to move next, and she's actually from Brookings and picked here, and there was a job opening, and I applied, got the position, and that was the last decade, I guess. So we really take an interdisciplinary approach to research. With my early training in physics and math, I try and use a lot of very quantitative approaches, so we do things like build microscopes in my lab. Our main biological goal is understanding cells of the immune system, and particularly a cell called the macrophage, or big eater. These are cells that are particularly geared for engulfing through the process of phagocytosis. They can engulf microorganisms or dead or dying cells and clear them from the body. And my lab is particularly focused on trying to understand the mechanisms of that engulfment process, how these macrophages recognize their target, and then how they actually build a phagosome around that target to kill and destroy and remove it from the body. So we use CRISPR-Cas9 as a way to really relate genetics, the genome, if you will, to the phenome or the function of the macrophage. So we've been developing approaches where we can use the CRISPR-Cas9 system to go in and disrupt genes in macrophages and ask which genes are required for phagocytosis, or what is their contribution to the phagocytosis process. So in our most recent work, we've been developing whole genome screens where we're actually going through and disrupting every gene in the mouse genome and then ranking them by function for either their contributions to forming a phagosome or suppressing the phagocytic process.
So a couple like rapid fire random questions. What's your favorite DNA nucleotide? Or do you have
boy. Do I have it's like asking what's your favorite color. I guess I like guanine. I don't know.
Right, that works.
It seems like it's green. I mean, it's like this, it's G, it's green, it's, I don't know.
Right.
Whereas A's and T's, I don't know, C is just sort of out there.
Makes sense. Gotcha.
I don't know.
And then, do you have a favorite protein or enzyme?
Oh boy. I don't know.
That's okay.
Yeah, that one's tough. I mean, I've got a bunch of different proteins that I like to think about. I like to think about the small g protein. I started my career studying the small GTPases. Right now we spend a lot of time thinking about antibodies and antibody receptors on the surfaces of phagocytes and that interaction.
What is something that most people don't know about you that you're willing to share?
I like to cook.
Do you have a favorite superhero?
I would say Iron Man.
Okay.
The high tech Iron.
Yeah, I approve. And a favorite type of candy?
I've always been a sucker for Snickers. I gotta say. So, one piece of advice I would have for any student that's interested in doing bioscience is one, you have to kind of become a linguist. There's a lot of terminology, but I think once you get past that initial phase of learning that terminology, you start to see what's happening underneath that. It's a lot more simple. So I would say, one, be ready to be a linguist and learn a lot of new terms for biology. The other one is be ready to integrate information from anywhere. Successful biologists, successful researchers are people that can draw on different disciplines and draw on different knowledge and be able to figure out all kinds of different problems. So be ready to innovate and dig in and use the knowledge that you can find.